Module 9, the peripheral nervous system. The sense of vision. Eye anatomy. The eye is an amazing sensory organ. Your eyes are able to take in light that is translated into amazing vistas and moving events that your brain stores and remembers. You are likely not outside as you are watching this, but if I ask you to think of a time you saw a rainbow in the sky, you probably can imagine in your mind what your eyes saw. Vision is one of the most complex yet most fascinating of the senses. Your eyes are so important that they are protected by two structures. The first, the palpebrae, are the eyelids. Eyelids and eyelashes protect the eyes from foreign particles that could cause damage. The eyelids also help to spread tears, and tears are the second mode of eye protection. They are produced by lacrimal glands in order to keep the eye moist and wash out foreign objects. If you are outside and a sudden wind blows dust into your eyes, what happens almost immediately? That's right, your eyes tear up. Your lacrimal glands produce tears to help wash out the dust particles that made it past your eyelashes and eyelids. Tears are also helpful in that they contain antibodies and a special enzyme called lysozyme that has an antibacterial effect. That is why your eyes rarely get infections. As your lacrimal glands produce tears, the excess fluid is drained away via four ducts called lacrimal canaliculi that lead to the nasal cavity. There is one duct on each palpebral. The opening of the lacrimal canaliculi are called punctae. If your eyes produce too many tears, they are unable to drain away, so they spill out of the eye and you cry. So your eyes can cry if there is an irritant such as dust blown into them, but they also cry as a response to felt emotions. And here's a fascinating fact about tears that you cry. The chemical makeup of tears produced from irritation is very different from the chemical makeup of tears produced by emotion. There are three coverings or tunics of tissue that surround the eye. The fibrous tunic is made up of the sclera and the cornea. The sclera is the white part of your eye that you can see. It is made of connective tissue that completely covers the eye except for the area where the cornea is. The sclera is firm and helps to maintain the eye's shape and keep the inside protected. It also provides a good spot for the eye muscles to attach. Now, the cornea is a clear area on the outside of the eye through which light can enter. Like the sclera, the cornea is also made of connective tissue, but its collagen fibers are thinner and there is less water present. This helps the cornea to be transparent. The cornea has another job as well. It helps provide focus for the light waves entering the eye. It has no moving focus ability, but its presence automatically refracts entering light so that it bends a bit before it reaches the lens. Now, the cornea and the part of the sclera are surrounded by an outer membrane called the conjunctiva. This is a very thin, transparent membrane made of epithelial tissue that covers the inside eyelid surface and the anterior part of the sclera. The conjunctiva's job is to help protect and lubricate the eye. That's what makes the exposed whites of your eyes to appear shiny. If you've ever had the infection called pink eye, it's caused by an inflammation of the conjunctiva due to a bacterial or viral infection. This doesn't happen often, though, because the smooth, slick surface of the conjunctiva makes it difficult for bacteria to adhere. That pretty much covers the fibrous tunic of the eye. The next layer of the eye is called the vascular tunic. This is the layer that has the most blood vessels in the eye, so it is well supplied with oxygen and nutrients. It's composed of three sections. The choroid is a layer of vascular tissue just beneath the sclera. It does not extend to the area of the eye near the cornea. At about 0.1 millimeters thick, it looks black because it has lots of cells that make the dark brown pigment called melanin. Remember that melanin is a pigment in your skin, too. Near the cornea, there is a ring-shaped ciliary body containing smooth muscles called ciliary muscles. These muscles help to control the shape of the lens. Now, this portion of the vascular tunic also has small projections called ciliary processes that surround the lens of the eye and are attached to the lens by suspensory ligaments. The third section of the vascular tunic is the iris. This is the colored part of the eye. If you have blue eyes, your iris is blue. If you have brown eyes, your iris is brown. In fact, eyes can appear blue, brown, green, gray, or a combination of these colors. The iris is made up of smooth muscle tissue. It surrounds the opening in the eye, which is called the pupil. The wider in diameter the pupil is, the more light can enter the eye. And that's the job of the iris. It contracts or relaxes to control the pupil's size, adjusting to control how much light enters. The sympathetic division of the autonomic nervous system is responsible for increasing the size of your pupils. You cannot consciously cause them to dilate. And the parasympathetic division of the autonomic nervous system controls decreasing the size of the pupils, causing them to constrict. And that covers the vascular tunic. Well, the last tunic of the eye is called the nervous tunic, and it is made up of the retina. The retina is about the size and thickness of a postage stamp, filled with blood vessels. If you've ever taken a photo of someone and noticed they had red dots in the center of their eyes, you are seeing the blood vessels of their retinas. The retina is the location of special cells that are called photoreceptors. They are rods and cones. You have about 120 million rods and 7 million cones in each eye. Now there's a spot on the retina called the optic disc where there are no rods or cones. This is because it's the spot where the blood vessels enter the eye and where the axons of the neurons of the retina exit the eye, joining the optic nerve. Because there are no photoreceptors at this spot, it behaves as a blind spot, not allowing us to see any light that is focused there. In the middle of the retina is a spot on the posterior of the eye called the macula lutea. It is only about 4 millimeters in diameter and appears as a yellow spot. Now, right in the middle of the macula lutea is a very small pit called the fovea centralis. This is usually the spot where light is focused, and it creates the sharpest image because the photoreceptor cells there that identify detailed colored vision are tightly packed. It is a good idea to understand a bit more about how our eye actually can perceive sight. We can do this by looking more closely at the histology of the retina. To orient you, this is the direction light comes in. Beginning at the deepest retinal layer, we have the pigmented retina. This is made up of one layer of epithelial cells that are full of melanin. This layer then is black in appearance. That means that when light hits it, it is all absorbed. And that's a good thing because this helps to increase the sharpness of the image coming in because light isn't reflected around the inside of the eye and hitting photoreceptors again. Excess light bouncing off the inside of the eye and hitting photoreceptors would cause those cells to create more action potentials, causing extra light information to make the image blurry. Now, though pigmented retina is helpful for vision, it is not necessary. There are some folks with albinism, meaning that their bodies are unable to produce melanin, and they are able to see, though they have compromised vision by bounce back of light. And when light bounces back off the retina, a reflex called nystagmus occurs. This reflex creates a shifting of the eyes caused by skeletal muscle to help them try to better focus on an object. Some animals don't have a black-colored choroid to absorb light. But though this gives them distorted images, the light that bounces back and hits the photoreceptors enables them to see better in the dark. Just before the pigmented retina are the rod and cone cells. These are the photoreceptor cells that respond to light. The rods give us vision that works in a nighttime dim light system. They are very sensitive to even small amounts of light, but they cannot distinguish the color of the light that hits them, so they're unable to send color information to the brain. Instead, all the information they send to the brain will be different shades of black, white, and gray. You can test this out by taking some pale-colored objects along with some white ones and go to a dimly lit area to view them. You will likely be unable to tell what color they are. They will all appear to be some shade of gray. Now, cones require a lot of light. They give us vision that works in a daytime, bright light system, and thus only work well where there's enough light available. Their job is to enhance how sharply an image is perceived. Additionally, cones are able to distinguish the wavelengths of light that hit the retina so they can identify color. Both rods and cones synapse with bipolar neurons that then synapse with ganglion cells. The ganglion cells then extend over the retina's surface to converge on the optic disc so that they can then become a part of the optic nerve. Now, they do not pass over the retina at the fovea centralis, where light is mainly focused. This is why the fovea centralis is a little bit thinner than the rest of the retina, and why it appears as a small pit. This is another reason why the fovea centralis is the best location for vision on the retina. Light doesn't have to pass through the axons of ganglion cells here. So because the fovea has a dense concentration of cones and has no axons passing over it, it is the source of best vision on the retina, and that is why the eye focuses light on that spot. The retina also has some association neurons that help regulate some signals before they make it to the brain. That means that the retina is a location of some pre-processing of information before it travels to the brain. Well, besides the tunics I've just talked about, there are two more components of the eye I need to mention. The vitreous humor is located in the large vitreous compartment in the rear of the eye. It is made up of a see-through gel-like protein that holds water. This protein attracts water so that it can form a gel. This substance is an extremely inert material, and it is thought that the vitreous humor material you have in your eyes right now is the same material you had when you were a newborn baby. The vitreous humor's job is to fill the eyeball, maintaining its spherical shape. It is completely clear material, but sometimes there are little objects that float around in it. These objects are called floaters. Floaters are strings of protein floating around in the vitreous humor that have come out of solution. They can be seen as tiny, faint strands when you look around. Another material of the eye is called aqueous humor. It is similar to cerebrospinal fluid makeup. You can find aqueous humor in the anterior and posterior chamber of the eye. It is made by the ciliary processes and flows through the chambers, draining back to the blood through a vein near the iris. Aqueous humor provides some automatic refraction to help the eye focus the light that is entering it. Additionally, it helps to maintain eye pressure, keeping the cornea slightly bulging outward. Believe it or not, pressure in the eye is very important. If there is too little, the retina would detach from the choroid. If there is too much, the neurons in the eye become damaged, and thus vision loss occurs. This situation is called glaucoma. Glaucoma is a loss of vision due to excess eye pressure. The aqueous humor is also helpful in that it provides nutrients to parts of the eye, such as the cornea, which has no blood vessels. Module 9, the peripheral nervous system. Experiment 9.2, cow's eye dissection. A cow eye is very similar in structure to the human eye, so by doing a dissection of the cow eye, we'll get a better understanding of all the parts of the eye structure. Before we begin doing the actual lab, you may want to go ahead and get some dissection gloves. And you can go to a local drugstore to get those. They'll either be blue or clear. You may want to get some that are latex-free if you have a latex allergy. But go ahead and it sometimes is a better idea to use gloves, even though it's not specified in your lab, unless you like the aroma of the preservative that's used in these specimens. It'll stay in your skin for a couple of days, and if you like that aroma, go right ahead. But I like to use the gloves. So let's go ahead and get our gloves on. And let's do a dissection. I also like to recommend that whatever table you're dissecting on, use a garbage bag as kind of a tablecloth. That helps protect the surface. And then when you're done with your dissection, you can take all the pieces that you want to dispose of and roll them up and place them right into the garbage. So I'm going to move all of my materials onto my tablecloth. And we're going to begin by taking a look at the exterior structures of the cow eye. So I'm going to remove it from the container. Now, thankfully, the preservative is not a very strong preservative, and it's not harmful to you, so you don't have to worry about that. But if you feel more comfortable doing this dissection outside, you certainly can do that. But you notice I'm doing it from an interior part of a room, so it's certainly not super strong. The first thing we want to take a look at with the cow eye itself and the external structures is the sclera. Now, the sclera is kind of hard to see here, but this is the anterior portion of the eye, and it is the tough outer layer of the eye right here. It's tough. It's when a person is alive or a cow is alive, this area is actually very clear, but because of the preservative, it's broken down some of the proteins and becomes a little more translucent, not completely clear. But we're looking at the sclera, and then on an important thing on the back side or the posterior side that I want to point out. Let me remove some of this tissue so you can see it a little bit on this specimen right here. You're looking for a tough extension coming out of the posterior side of the eye. And it's right here, this thing right here. This is the optic nerve. The optic nerve goes directly to the brain and sends the signals that the eye receives straight to the brain. So we'll take a closer look at that once we get inside of the eye as well. But I want you to notice this from the outside. You might also be able to see some of these darker, tougher bits of material. This right here, these are muscles. And that the muscles are necessary, of course, to move the eye from one direction to another. And so you can see another example. There's another muscle piece right here from the posterior side. So that is we're back to the anterior portion of the eye. And the last thing I want to point out is the cornea. The cornea is, again, perfectly clear in life, but it's a blue-gray color right now due to the preservative that it's been kept in. And we are also going to look at the conjunctiva, which is directly behind that. Now, this is the conjunctiva in my specimen. Let me get my tweezers to kind of pick it up a little bit so you can see that. You may have a specimen where the conjunctiva is darker. It either is going to, and so again, that would be as a result of the preservative changing the color or denaturing those proteins. But this right here, you can see me pulling. This right here is the conjunctiva of the eye. All right, well, we're going to now begin to dissect the eye and get a closer look at what's going on inside. So the way to do that is to begin cutting, and I like to use my scalpel to start the incision, and then we'll use the scissors to go all the way around. We're going to start just past that conjunctiva and just make a tiny cut to begin with. And you don't want to go very deep if possible because we don't want to damage the interior portions of the eye. So let me see if I can get started here. Might be better to use my scissors to get started. And you can see the eye changing shape a little bit as I'm putting pressure on it. It's because it's filled with fluid, and that helps keep the structure of the eye rounded. So there will be some fluid loss when we get into here. Well, there we go. All right. And you want to do just a circumference all the way around with your scissors. When you cut with scissors and doing a dissection, it's always good to pull up with that part that's down inside the specimen, because if you go down with it, you see how you could possibly get into material that's down underneath where you're cutting. So always kind of pull up gently a little bit with that part that goes into your specimen. And again, we're doing a circumference. Just open it up all the way. And this will enable us to look at the anterior portion of the eye from the inside, as well as the posterior portion. I'll try to keep my fingers out of the way here as best as possible, so you can get a good look. I'm cutting where some of those muscles are, and you can see how it's a little tougher. Just be patient as you do this, because if you go too fast, you may do some damage and won't be able to see as much. Now you can see some of the material is coming out of the eye as I'm cutting through, and that's okay. We don't need to worry about that. I'm cutting through some very tough muscle. All right, and there we go. Okay, you can see here now, this part right here is the anterior portion of the eye. Let me just show you again. Actually, no, it's not. We move this. Let me move this back over here to this side. This goes in this side. There we go. Now, there's the anterior portion of the eye, and this is the posterior portion where you can see the optic nerve coming out right here. Okay, so just to orient you again, anterior side, posterior side. All right, and so at this point we're going to look at the anterior portion. So I'm going to move the posterior portion over just a bit so we can focus on the anterior portion right here. And we want to take a look from the inside. First I'm going to remove this thing right here, which is the lens. And you can see how the lens is surrounded by fluid. That fluid helps protect it, keeps it suspended, and provides that pressure to keep the material, keep the eye in its shape. We want the eye to have that nice round shape. We're going to begin by looking at the pupil here. Let me use my pointer for you to see. This opening right here in the center is the pupil. So you can kind of see a bluish gray line right here. in humans, the pupil is round in shape. Cow pupils are more oblong in shape. So that's one difference. But this is the pupil. It's the hole that can enlarge or contract to regulate the light entry. And then the iris is the thin black tissue that surrounds the pupil. So right here, then, is the iris, this black material. Let's see if I can pick that up for you to see. This is the iris that surrounds the pupil. And around the iris, you're going to see what looks like fibrous material that almost sunburst in shape, as I'm kind of trailing along with it with my pointer. This fibrous material here, this is the ciliary body. They're in radial formation. And the ciliary body is what attaches to the lens via suspensory ligaments. And that has, those muscles that it has are what will change the shape of the lens. So if you can imagine this lens right here, back in this spot, you can hopefully see the circle of muscle starting at the edges right here, and then the ciliary body right here. The suspensory ligaments are a little more challenging to see because they're clear and they're very tightly attached to the ciliary body. Page. Okay, so we're done looking at the anterior portion of the eye. I want to point out the lens to you, though, one more time. The lens is normally spherical in shape. It's a little bit oblong in shape right now, again, because this has been preserved. But the lens can be changed based on the pulling, the contracting or relaxing of the ciliary body, and that helps it to focus the light onto the retina of the eye. So I'm going to move this anterior portion of the eye over, and we're now going to focus on the posterior portion of the eye. The first thing we want to look at, the posterior portion, is the retina. Now, the retina in this case is very difficult to see the way it originally looked. It's this tan-colored material that's kind of collected into a glob. Normally, the retina is spread out completely over this portion of the eye, just completely covering this portion of the eye. But because the fluid is moved, and this, of course, again, is a preserved specimen, it's kind of collapsed on itself. What keeps the retina attached is the pressure of all this fluid that is inside the eye. So again, normally it's completely spread along the back posterior portion of the eye to receive light as it comes in. But I want you to notice also, if I move the retina around a little bit, it's attached at one spot. Can you see? Ooh, this is a better angle right here. You see where it's attached right there? That's where all the nerves, all the neuron extends, the neurons of the retina extend and go through this point out the back to the optic nerve. So that is why this has to be attached, because all of the neurons are connected through that optic nerve going to the back. And so this spot right here is called the optic disc. That's where the optic nerve exits the eye. It's also your blind spot where you cannot see, because that's where all those neurons exit. All right, now the stiffer, thin middle layer is called the choroid, and it can be pulled away from the sclera at the edge of this dissection. The choroid is this iridescent color in cattle. In humans, it's all black, but this beautiful bluish iridescent color is for animals that are more nocturnal-like or dusk and dawn active. It helps them to see better when there's not as much light. But humans, it's all black. And so let's see if I can pull that away, and you can see how it easily comes. It's just a thin layer underneath the retina, like that. So you can see that. Okay. All right, so that is the choroid. And I want to pull more of the choroid so you can see the sclera again. And the sclera means hard or tough. So let me do that. And while I'm holding it back, can you see how it is indeed tough and hard? When I push it in, oops, get that out of the way. When I push in, it bounces back. When I push in, it bounces back. That sclera helps the eye to maintain its shape. It's nice and tough and protected so that it maintains that spherical shape. Now we want to look again at the lens and the suspensory ligaments and the vitreous humor. Okay, when in life, in living organisms, the lens is softer. I'm going to remove it actually from that anterior portion. Come on, lens. There we go. All right, so clear out my spot here. So this is the lens. In living organisms, the lens is very pliable and soft, and it's completely clear, like I mentioned earlier. Again, it is not clear here because of the preservatives that are used. So it's kind of clouded. And so it's marble shaped, and it's inside the lens capsule, which is a clear sac that holds the lens and protects it. All right, the black pigment around the capsule, which is when this was back here, these are where the suspensory ligaments are, and I pointed those out to you as already, where the suspensory ligaments are that hold it to the ciliary muscles. And so we also are noticing, if you've not noticed already, this goopy stuff. You see this goopy stuff that's the lens is around or around it? That's called vitreous humor. Vitreous humor is part of the fluid that is inside the eye to help hold its shape, and it's kind of gel-shaped. The fluid, the liquidy, watery fluid, is aqueous humor. So all the stuff that poured out when I cut open this eye, the liquidy stuff or watery stuff was aqueous humor, and this thicker material, this gel-like material, is the vitreous humor. All right, and so the final thing we want to do is we want to take a look at the inside of the lens. We want to cut it in two. And so I'm going to try to hold it steady and slice it. Let's try that. There we go. All right, and what you're going to see is the lens is made of concentric circles around and around and around, and it's a little bit difficult to see from this vantage point. Much easier to see if you're looking at it in person. So again, I encourage you to do this on your own, but it's made of concentric circles and layers and layers and layers of tissue. All right, the layers of this tissue, those are epithelial cells, and again, they're avascular, so there's no veins in this area feeding blood to this area, but this is our epithelial tissue, kind of layered like the layers of an onion might be. Okay, well, we have done a good orientation of the cow eye. I hope you have learned a little bit more, and can you see the similarities to the diagrams that you were looking at? That's something that's very important to know. By doing this, you're helping yourself to better be oriented about the different features of the eye by seeing it yourself this way versus just relying on a diagram. So I encourage you to do this lab and enjoy really understanding better the fascinating structure of the eye.